Hello, everyone, and welcome to this joint webinar sponsored by the International Wheat Genome Sequencing Consortium and the Phytobiomes Alliance. My name is Dusty Gallagher. I'm the Phytobiomes Alliance Deputy Director, and I welcome all of you to this very special webinar. To get started, I'd like to share a few slides, but want to really leave as much time for our special guest as possible. So just a few words about the International Wheat Genome Sequencing Consortium. This was established in 2005 by a group of wheat growers, plant scientists, public and private breeders. And the idea is to accelerate wheat improvement. So since the time of the consortium, they have produced the reference sequence of bred wheat in 2017. And then the latest version of assembly and annotation is the IWGSC RefSec version 2.1. And the vision of the consortium is to enhance breeding through an increased understanding of the molecular basis of traits and their allelic diversity. We'd like to thank our sponsors of the IWGSC. Without their contributions, we wouldn't be as successful as we've been since 2005. The International Alliance for Phytobiomes Research is a nonprofit pre-competitive research consortium that is made up of industry, academia, and government. And our goal is to create a paradigm shift in agricultural research and production. And so our vision is that by 2050, all the farmers will have the ability to use predictive and prescriptive analytics based on the geophysical and biological conditions for determining the best combination of crops, management practices, and inputs for a specific site in any given year. And then our strategy, what we're working on, is to identify the gaps in research tools and technology and develop projects that will address those gaps. So just quickly, the concept of the phytobiome is that it's plant-based agriculture in a particular substrate. And so for this visual, we're thinking soil, but that can be many different. It could be a hydroponic system. It could be a contained system. It could be a pasture, livestock. And then we're looking at the complex systems around that phytobiomes, thinking about all the different elements that influence the production of that plant in that specific site. And then we're looking at the gaps between all of those systems, knowing that all of this is influenced by management practices. We couldn't do what we do without the sponsors of the Phytobiomes Alliance, and so we thank them for their support. And then I wanted to mention a couple of upcoming events for each of the organizations. For the Phytobiomes Alliance, we're currently planning the 2024 International Phytobiomes Conference, which will be 8 through 10 in October in St. Louis, Missouri. Registration for the conference and ability to submit abstracts is currently available and open. And can be accessed through the phytobiomesconference.org website. And then for the IWGSC, we have an upcoming webinar in April, just a little under a month from now, and we will be focusing on the grain genes, the centralized nexus for small grains data and communities. So be sure that you register for both of those events. And just a couple of housekeeping items for our webinar today. This webinar is being recorded and will be posted on both the IWGSC and Phytobiomes Alliance and USCCN YouTube channels later today. So if you want to go back, rewatch, then that will be available to you. And then our modes of engagement for today are, we would like for you to use the Q&A panel to submit your questions. We will have some time at the end of the presentation for a Q&A discussion. So please use that Q&A panel for that purpose only. The chat panel is there, and it is to be used to contact and see messages from the organizers. So that that tool is only to communicate with the organizers of the webinar. And then there's a handout section that you can see some of the PDFs from today's presentations. This presentation that I'm giving right now will be included. And then we will later on our websites post the main presentation for today's webinar. So make sure that you're signed up on the mailing lists to receive news about the Phytobiomes Alliance and IWGSC for more information about our upcoming events, webinars, etc. So without further ado, we are very honored to have Professor Dr. Schurr with us today, who is going to be talking about plant phenotyping, technologies, and targets. So I turn the podium over to Dr. Schurr. We welcome you and look forward to your presentation.
So thanks very much, Dusty, for the very nice introduction, and also thanks very much to the opportunity to give this presentation here, this webinar today. I'm trying to show you a little bit of technologies and targets and present developments in plant phenotyping. It can obviously, because the field is extremely wide, only be scratching the surface, but nevertheless, I hope that there are some points in there which you can take with you, and then also, as has been said just now, if you have further questions we can actually discuss this in the discussion, or as you see on the first slide here, you can also contact me directly or any of my colleagues. So this is a presentation which I present today, but for a whole community of people, not only the people which are mentioned on the list here, but also many people from all around the world contributing to this plant phenotyping technology development targets in the last, I would say, 25, 30 years when we're working on that. I'm mostly covering technologies today, less what has been measured with things, but that might be a thing to discuss further in the context. So a lot of the points which have been raised just now, for microbiome to stresses to agronomics, but breeding, many of them require plant phenotyping. So let me start very general into the presentation. If there are many challenges out there for agriculture, and I don't think we have to go into more details here in this audience with what is there. We have to increase yields, we have to increase the quality. There's a lot of adaptation for different crops, for different habitats. On the other hand, we have to reduce inputs with respect to fertilizers, chemicals, but also water consumption. And we have two main measures to do that. One is breeding and the other one is management. And in both cases, what we require to do that is quantitative data, which are innovative, giving us innovative insights and provide solutions for breeding as well as crop management. And therefore, from our perspective, we are essentially doing that in order to do three major parts to deliver into these challenges towards the agriculture in the 21st century. On the one hand, we try to understand what plants are actually doing, because with phenotyping, we can get, with the modern types of phenotyping, we can get very detailed information, and I will show you some things, detailed information in space and time and new mechanisms. Secondly, obviously, we have to design new crops. We have to design new production systems. I will also show you a little bit of that, but this is obviously based on quantitative information. And last but not least, it's also about managing crops in the right way. So there are sensors, there are optical information, but also a lot of, to the entire spectrum of information, sensors which we do, optical sensors, but point sensors, but as well as imaging sensors. We do can gain a lot of information which we can directly use to manage and build into management systems. So therefore, phenotyping is part of an entire world of digital innovation in agriculture. And I just wanted to put this one in here from an excellence cluster, which we are also collaborating very strongly here together with the University of Bonn, where I just want to show you the diversity, the need to interact. So the first one is that obviously there is a whole suite of novel digital or often digital technologies There's a lot of machine learning, there's KI, there's a lot of stuff in there. There's a lot of monitoring things, devices, robots, monitoring flying robots, driving robots. There's also intervention, which is obviously requiring the information on the ground. At the same time, we need to have the understanding of plant, soil, environment in a whole. So this is where the phenotyping comes in. This is where plant-soil interaction comes in, where the microbe comes in, where the whole suite of biological information comes in. And this has to be combined. And I think that's a very crucial thing over the last, as I said, several decades, which we're working on that we have this tight interaction between the technologies and the understanding. And this is a kind of dialogue between these levels. Very importantly, on the other hand, to really get an impact is that we are integrating these kind of technologies and understanding into a wider community, meaning economic impact for the farmer, economic impact for breeding, economic and also impact for the environment. So therefore, we need to build this whole phenotyping community from technologies and understanding into a wider picture. And that's actually one of the things which we also do in this excellence cluster where we do this. On top of this perspective, we also need to make sure that this is not only about in certain dimensions. This includes different technology scales as well. This includes things linking to remote sensing. I will actually only touch this very briefly in this presentation, but we also can take remote sensing into the bigger picture. And at the same time, it's not only about expensive gadgets, about getting access to them and things like that, but it's also a whole suite of things happening today on what we would call affordable digital technologies. So everybody has smartphones in our hands, and the technologies, the sensors, and the compute power which is in there is opening a whole suite of new opportunities in the future, which we also use partly already in plant phenotyping. I still think there's much more coming in there, not only from the smartphone level, but also from all these other small computers which are available now at our hands. If we look back into the plant, then the digital information which we would like to have is requiring a whole large number of different traits, a large number of different information which we want to deliver. And actually from seed to seed. So it's through the entire life cycle. It includes seed traits, includes also sowing practices and germination, key process in vegetative phase. The generative phase itself is a very important one, harvest. So essentially from seed to seed, that's the whole life cycle which we would like to have quantitative information on. And in our context, we also put this into a joint direct phenotyping databases, because as we know, just getting information and getting data from just collecting them without bringing them into a unified data collection is not the way we should do this today anymore, because there's an increasing number and large number of data which is out there. I will come to that at the very end, a short perspective. But this requires then to also have tools in hand which go through the entire life cycle of a plant. So we have seeds, we work on seed stuff, we work on seed establishment. So in large scale, we also go in vegetative growth, and we go into yield and harvesting products, which we can go into very detailed aspects. And as you can also see from this picture here, we have to work on a wide range of different species, which is very different to phenotyping in the medical domain, where obviously there's a lot of technologies out there which are also doing phenotyping, but they have the big advantage that they have just a single species, and we are working with many different species. So that's an additional complication which we have. And I would like to go through this seminar through the life cycle of a crop and what kind of technologies and opportunities we have in there in order to get quantitative information. As I said, just scratching the surface, there's much more, and I'm obviously mostly focusing on what we have here on hand here, but there's also a lot of stuff which happens outside. So let's start with seeds. So why is seed phenotyping? What is the starting point for that? And for us, the starting point, because it was actually to optimize the phenotyping pipeline itself. Because if you run your expensive phenotyping pipelines with a uncharacterized batch of seeds, then you introduce your statistical variance without having any meaning in that. So therefore, we started to do some of our seed phenotyping initially from that side. At the same situation, we actually then realized that there was quite a large number of people asking about seed physiology. So seed properties, the germination, and things like that, where this in itself became for us also a field of phenotyping, where seed physiologists wanted to know more details about what actually happens in the seeds and how seed properties develop. A third one, again, a very different approach, is to characterize the germplasm. So we have a lot of seed banks out there. Seed banks not having many millions of batches of bags where some seeds are in there, but how can we characterize all of them, and what are the main characteristics of the seeds? And therefore, we also have one stream in our institute here where we are looking into characterization of germplasm in seed banks, because they require a very different approach relative to the two parts which I mentioned on the upper side of that slide, phenotyping pipeline and seed physiology. And last but not least, there's a commercial part in there, seed testing for seed industry. There is a lot of activities there, obviously, for standard tests. But a lot of these standard tests could actually be improved significantly by getting quantitative data from modern phenotyping technologies, which we are also working on to some extent. So what kind of seed phenotyping is out there? There is the classical ones, bulk seed properties. The bulk seed properties where you have 1,000 kernel weights, for example, and there are these machines which you can buy, which is a very traditional way how to measure seeds. There are other things which came up in the last years where you measure bulk seeds, but you can, in the bulk sample, you can measure the properties of individual seeds. So this is, for example, one example from CNR, where you have a number of different seeds there, but you can measure seeds of properties of individual seeds, but you can't collect and take them afterwards and sort them. Therefore, we also set up a seed sorting system where we actually work with individual seeds, where we can pick them and place them and sort them. And there are obviously extreme detailed seed phenotyping with CTs, with MRIs, where you actually see inside the seed itself. You can actually have three-dimensional scans using CTs, using MRIs now today, with, for example, even Arabidopsis, where you can fly through the seed and where you can get this information. We also ourselves work on that to, for example, get information between storage and embryos and how they are working. This also shows that there's a wide range of different technologies, but it also is a good indication for, which is a principle which you can find in many contexts, that there is a trade-off between throughput and information. If you go to the very high information side, the throughput is low. If you go to a high throughput side, the information is usually quite low. So this is a kind of trade-off which you often have to work with, and that's obviously a thing where even with automation you can get a little bit shifting things around, but still it's an issue where which is very common. So just to give you some examples here, so a system which we have here now, which we're actually working together with Fraunhofer, is a X-ray CT, which we use to characterize seeds, individual seeds, as well as seed lots. And on the right, and I hope that the video is broadcasted, you can see how a CT is actually looking there. We are flying through a batch of a sample of seeds. And by looking at these individual seeds and doing image analysis with that, we can then gain that kind of block information. But we can also segment individual seeds out of there and take information from individual seeds in that batch, and therefore get additional information. This picture here shows that we can also do virtual sections, and this gives us the information in the segmented sample that we can then actually measure for individual seeds in the bulk what the properties of these systems are, of the seeds are. Another option is to use this kind of phenoseeder, a system which we also built now several years ago. It's a robot the robot picks individual seeds and puts them in different measuring modes. And you can see that we do, for example, three-dimensional reconstruction. We do weighing. We do weighing in different properties, large, small. And we have tested a large number of different seeds and worked with a large number of different seeds in that context. And you see some of the species there. There are more species in the meantime. Traits which are measured: projected area, volume, mass, density, color, 3D shapes, all of these kind of things are possible. We actually combine that now also with the CT so that we can get individual seeds from the size of maize down to Arabidopsis, where we can actually get three-dimensional information as well from individual seeds, which are usually treated then by this phenoseeder. What can you do with it? You can, on the one hand, look at the bulk properties. Also with this phenoseeder, you can look at the bulk properties here, and you can see a distribution of seed volume, seed mass, seed density, seed median. And this is what I mentioned at the beginning. So if you go into your seed phenotyping pipelines with this kind of diversity of seeds from small to big, then it's not unlikely that you're increasing the diversity and the statistical error, which is just coming from a diversity of the seed lot which you put in, compared to a more defined seed material. So we can, and with the phenoseeder, we can actually take individual seeds from different parts of the distribution and then work with these individual seeds. So all the plants essentially which we have in our phenotyping pipelines have gone through the phenoseeder. So we know from each plant which we have in our phenotyping pipeline what the initial seed quality was, where it came from. And we can also, for example, take samples where we say we are reducing the variability of the seed lot by just taking the median of that and then thereby reduce significantly the statistical variability of the seeds in order to go in there. Seeds on the one hand coming into the pipeline, on the other hand, obviously seeds are also growing, and seeds are interesting in order to study as a physiological system and in an intact plant. So in this case on the left, and I'll come back to that kind of technology later on in more details, we have MRIs which are large. In this specific case, this is our largest MRI system which we have, which allows us to measure plants up to 4.50 meters height. This includes obviously also things like maize, and on the right you see this greenish color, which is false color obviously of MRI, but you can see that we can see the seeds in there. We can do this non-invasively, and we can see how the development of individual seeds in that cob can work over time. And then obviously also can take samples from there with a much more detailed information than if you would just cut the cob and take it somewhere. So this is an opportunity to get seed structures in vivo, but we can also use, beyond the imaging, we have also sensors which are doing, in this case, MRI. MRI has an opportunity at certain modes to measure not only the water or the watery content, which is on the right side in this picture here, the liquid part, so the proton density liquid, so the blue one, but you can also measure the protons which are in solids. And within the same sensor, we can measure in this specific case actually both at the same time, the protons in solids and the protons in liquids. And what you can actually see on this slide, on this graph here, is the import of carbon, the import of solid material, the import of water, and how they are differentiating over time at a temporal resolution of down to 10 minutes or less if you want. And in this case, you can see the diurnal variations of the input of water and carbon into the seed pod here. So these are the kind of opportunities which are there in this specific case. So measuring, sorting, getting information about seeds in vivo, how they are developing, how the harvest is developing, and on the other hand, also these kind of sensors which give us a very detailed information about influx and even efflux of material. So that's just scratching a little bit the surface of what can be done with seed phenotyping. I now want to switch over to phenotyping of roots and shoots, because that's obviously for the vegetative phase the more important one. And let's start with the root part. Roots are living in a very special environment, and this is still one of the most obvious pictures from Michel Watt on the right side, where you can see a micrograph, how intimate the interaction between the root and the soil actually is. And the roots are living in this very special environment, which are completely different with respect to the spatial patterns, the temporal patterns, the chemical structured environment which they are living in, and the mechanical properties. They are completely different from above ground. At the same time, what we often do is we're studying them in these kind of artificial conditions, conditions which definitely are not mimicking the situation in the field, and therefore the information which we get from these experiments have a certain limitation. I don't say they are useless, but there are certain limitations, especially if you look into the efficiency of root systems in structure and function in the soil. So what does this mean? This means that we have to somehow work towards a pipeline of root phenotyping, and we have done quite a lot of them in different dimensions, from using different technologies. We have a PET system, positron emission tomography system, which I'll come to in a minute. But then we also go into high throughput systems. We have a number of high throughput systems in the greenhouse, which are also looking at roots in the soil. And we also work towards a bigger perspective. So also from root architecture at plant level to plant community level, where the roots are also interacting below ground. And then obviously still there's on the fields level, there still needs to be destructive sampling. It's from time to time, but we are very strongly working, trying to get also root sampling, root phenotyping on the field by non-invasive technologies. That's still ongoing. It's not there, but we have, I think, good examples how to do that. So let me start with the more detailed or very detailed information. So real deep phenotyping on the spatial and temporal dynamics of roots. And in our case, we're doing that with tomographic systems. For exactly the reason which I mentioned at the beginning, if we take the root out of the soil context, we completely lose the geometry, we lose the interaction, we lose the physical and chemical limitations which are given to the root, and where the root is actually best performing. And therefore, we are looking into all the technologies which are tomographic systems, MRI, PET, CT. There's quite a lot of different technologies there. So ultrasound. So there's a wide technology level which is available, often from other background, but in our case, very much useful also for plants. In many cases, we need to define and develop specific technologies for plants, because, for example, in MRI, which all of you, or hopefully not too many of you, have actually been in there, but most of us have been in there. As a patient, you have to lay down horizontal. Plants don't like to do that. Therefore, our MRIs are vertical, because plants are growing vertical, not horizontal. And therefore, we have to build completely new systems in many of that background. Also with the PETs, positron emission tomography is a system which is well known from medical, but we need to have special treatments for that. In our specific case, we are working with a short-lived isotope of carbon called carbon-11. Carbon-11 has a half-time of 20 minutes, and 20 minutes half-time you definitely can't buy somewhere. Therefore, we have to have our own cyclotron, and we have a cyclotron here for plant research in our institute, which we use to produce C11 CO2, which we again can watch how it works, how it runs through the plant in lifetime with the PhenoPet, for example. The combination of both gives us the opportunity to see the structure with the MRI and the carbon transport, meaning the function and the activity of the roots, using the PET. And the 3D reconstruction gives us the opportunity to say which root is active and which root is not active, and where is the carbon going and how is the root growing, and where's the carbon going when the root is growing. So all of these kind of things are possible. An example for that is this one here, or a number of examples. So on the left, you see that we can do short-term dynamics. This is a root system where when I start the video, you can see that this is a carbon flowing into that root system. That's actually in real, it's a three-dimensional root system. And you can see how the carbon actually is flowing in there. This is over a sequence of several minutes, half an hour to 2 hours. That's the maximum which you can do with this kind of resolution. But you can clearly see that there are certain spots which have more carbon, get more carbon allocated, while others are less. And this is on the one hand, the root tips, which you can see here, these small tips down here. But there are also possibilities to measure, for example, nodules in legumes, how they are actually activated, and which one are activated, which one are not. So there's a large heterogeneity, and we can see the full spatial and temporal dynamics of that. We can also go to day-to-day activities and see how the carbons, how the root system is actually developing. In this case, you can see over days, repeatedly from the same specimen, from the same plant, you can see how this actually is developing over time. Each individual root can be picked, and you can do that. And on the right side, you can see that this can also be done quantitatively, non-invasively. All of these are root systems in soil. On the right side, you see diameters, this depicted in colors, so different diameters giving different functions of the root, and all of this can be combined in space and time. Well, this gives us a lot of opportunities, but it also is a technology which requires time and which is a deep phenotyping information, and therefore it has a high information density but low frequency. Therefore, we started some years ago to build rhizotrons and to work with rhizotrons, and we have a range of different rhizotron systems which we have established over the years. The one which we're using today is a fully automated rhizotron which can run a rhizotron greenhouse, which looks like over what you can see in this greenish screen here. This covers between 850 and 1,000 rhizotrons. We're using this kind of robots, which you can see in the video here, to transport the rhizotrons to the imaging boxes. You can see the imaging boxes here on the right. It's a fully automated logistics system, how to transport them there. You can see how they are transported to the imaging systems. Then the rhizotrons go into the imaging boxes. They are pulled in there, and then the cameras from different sites, below ground, above ground, are taking pictures, different sections of different qualities of pictures, to give information which we actually work with later on in order to do that. It's extremely high flexible system. We can work with 1,000 different experiments or with all of them in one experiment, and we can also mix experiments in the same system. There are many traits which we measure. This is just a short coverage of shoot traits plus shoot traits. So this is obviously taking in images and information from above ground and below ground. And we are permanently adding new information in that level to give information there. Before I go over to the next level, I just want to continue a little bit still on the non-invasive imaging, because there are some new things coming up which we have worked in the past year. And one of them is to look at computer tomography on wood and the xylem internal structures of the stem. And this is a picture which I think is really nicely illustrating how the three-dimensional structure can be used in order to follow individual xylem vessels, and how they are actually linked and reconnected inside a connection of the stem. This is also three-dimensional, as you can see here. It can swap, and we can do this actually also repeatedly and see also, combining these CT pictures with MRI, how the water flux in the individual parts is actually taking place. Another option which we have is we can look into MRI of fruits. So this is a pear, and this is a mandarin, and then we have also more detailed information about uptake of water into, for example, cherries. So this is a cherry MRI picture, structure inside, and this is how actually water goes in there when droplets are on there. And there's a whole range of different things. One thing which we are actually very proud of is that we also can do images of MRI in the field. So this is an orchard in an apple orchard. And in this video here, you can see how during a drought, the individual embolism takes place in the stem of these orchards. And we can also obviously see how the refilling of these xylem vessels actually happens. This immediately leads me over to the next level, phenotyping in the field. Phenotyping in the field is obviously the thing where we want to go for, because in the end, the plants have to perform in the field. And there's a whole, again, dimension which have to be brought together, and none of them is flawless. None of them is the key and the golden bullet, but all of them have to be combined. And that's the reason why we also go into these platforms and the networks which we have there. In our field phenotyping concept, there's obviously, we have to define which traits we would like to look at. And this is structural traits on the one hand, but also functional traits like water relations or photosynthesis. There's a whole suite of sensors for different plant traits, plant performance and quantification, from stereo imaging or multi-camera imaging, laser-induced fluorescent imaging, sun-induced fluorescence, plant eyes, a LiDAR system. So there's a wide range of different sensors, and this is by far not everything which is there. Obviously, in the field, you need specific positioning systems, and in the next slide, I will show you a number of different positioning systems. There's a range of specific experiments which are also needed in order to run phenotyping machines in the field. And obviously, environmental sensors are also very important, because in the field, we have the very important element of the environment, and we have to measure that non-invasively, continuously, repeatedly, with essentially the same structural and spatial resolution as the plant properties itself. There's a wide range of different positioning systems. There are positioning systems with drones, fixed wings, octocopters. There's the field snake, which is our largest machine, which has a 22 meters, which is 22-meter gadget, which is running over, in this case, specifically the phase system, which you can also see on the right down here. The phase system is a specific experiment where we actually grow plants at high CO2, and therefore we needed to have this ranging bridge positioning system. There's also automated, fully automated systems. There are, but also very useful push bikes which are dedicated for that. And we also have now a new transportable bridge system, the field weasel, which actually can go to different breeders and farmers and being tested in different backgrounds at the moment. And we also do this in different scales. As I mentioned before, we can go from, in this specific case, for photosynthesis from individual leaves, or even smaller, obviously in the greenhouse, in the greenhouse and growth chambers, to mini plots, to systems in the field, to plot size, then complete fields, then going to larger scales with still a resolution of 1 square meter, but flying over entire fields. And then the biggest scale which we are running at the moment is sun-induced fluorescence from space. This is a project, ESA satellite, which will be launched hopefully finally now in 2026. And then we'll give photosynthesis information for the first time from space using sun-induced fluorescence at a resolution which is 300 square meters globally within 3 days. And that's a very useful information, but there will be more detailed information, commercial satellites coming up later on. This is just to illustrate a little bit the bigger picture using satellites, using airplanes. And I don't want to go into details of the sun-induced fluorescence now, but it gives you the information how photosynthesis is distributed in space in a whole, in a field arrangement. So what you can see here is different setups, and you can essentially thereby directly measure with sun-induced fluorescence the photosynthesis on the ground on the basis of an airplane. As I mentioned, we also go for specific experiment. I also already mentioned the phase, because we want to simulate future CO2 concentrations, in this case in the phase system. And then obviously the measurement of the genotypes which are exposed to this high CO2 are very important. And it's an expensive experiment because of the CO2 and also the equipment which is run otherwise in there. And therefore, we want to get as much information as possible using these phenotyping devices, which you can see here. This is the large one. This includes the phase system itself. It's including the measurement system, but the field snake, how we call this whole device here, is a semi-automated positioning system, which is running over these rings and thereby measure with a number of sensors, which we can mount on this system here in the different rings which we have here. And down here, you can see the 4 rings which we have, which we can use in order to measure these different genotypes with many different aspects in the field. These systems can also be used either the field snake or also flying systems in order to have very applied experiments, very applied data. For example, for measuring cauliflower harvest time, or for measuring beans or sugar or any other vegetables as well. There's a large number of activities there for different analysis there. Don't have the time to go into detail for that now. But I would like to go over to also show you a new system which we have, which is actually an element into a new production system. You probably have heard about agri-PV, is agro-photovoltaics. So photovoltaics on the top of a field or in vertical systems next to crops. And this gives a completely different opportunity now to actually also do crop management, to do crop measurements. And there's a whole suite of different things which you can do in this context. And I don't want to talk about agri-PV now too much, but we are rolling out quite a lot of large systems now here in Germany. There's also globally, we are involved in a number of a global network in agri-PV with different benefits where the PV has actually benefits for the crops. But in the context of phenotyping, this also gives an opportunity here. And this is our experimental station of the agri-PV, where we have a rail system which is running on the field. And this is kind of camera car, which is running in this rail system, hanging down from the rail system. And down here, you see how the camera car, the tracks of this camera car and how it actually can go through these different systems. This can obviously not only be done for the agri-PV systems, but also for other systems. And we can then get information about biomass, 3D structures, but all the other things which we have measured before or have shown you before in the field sites. A little bit looking at the time. So the data systems are very important. At the current status is that there's a lot of objects which are out there. There's a lot of findable and a lot of measurements. This is the current status in the good things where we already have very good information. And this is also a picture which I took from Emphasis, the European infrastructure for plant phenotyping, which we are developing over the last years. And but what we really would like to have is a real reusable and refinable, accessible and reusable data because of all this different information. And Emphasis has set out very specifically in order to develop these kind of data information systems as well. This is local data information systems, but also trying to integrate them on a bigger picture. And therefore, things like MIAP, for example, so standard descriptions need to be done and need to be checked. And all the data models which are behind there need to be fitted into the data acquisition part and then also for the reuse part of crops. So there's a lot of standards developing there, community standards, depending on what kind of crop you're working, but also needing to combine with the phenotyping devices. There's a lot of input there. And we also have developing a lot of activities towards repositories, databases, and how they should optimally work together in the web services. This is also obviously then a thing which has to happen on a larger scale in collaborations. Therefore, we started to interact there between the European Plant Phenotyping Infrastructure, Emphasis, and Alex here for the bioinformatics, and a number of national institutes involved in there, because this can only be useful if it's done in a community level. Just running out of time a little bit, so I just jump over to the phenotyping networks because that's important to not only use this kind of gears and gadgets for yourself, but also using the technology development, the availability of the systems, providing access to them. And therefore, our developments in the past were that we also had European projects which provided access to these systems. The first one was to a limited number because we first wanted to test how this works. And then we increased that number. And this was an extremely successful way to provide access for researchers to these state-of-the-art phenotyping devices in order to also make a lot of publications out of a relatively small number of money. And this evolved into a whole picture of national infrastructures, and there are obviously also national infrastructure in the US, and APPN, for example, and others. And there, on the other hand, then we have this regional infrastructures bringing things together, the European Plant Phenotyping Network, this kind of projects. And now, therefore, we are working on since 2016 now towards a S3, a European Strategy Forum for Research Infrastructures concept for emphasis in order to make a long-term and stable organization, which is integrating and developing all this operation at a European level. And that's ongoing. And the targets is to develop this infrastructure of instrumented facilities, to develop linked data acquisition at a European level, and to develop and evaluate this also novel technologies. This is just a short glimpse here, and I'm actually cutting this down very, very strongly now. We have 5 different levels where we are working on: controlled environment, lean field, intensive field and modeling, and data and computational services. And the target of emphasis is to build this over the next years. So we are now in the implementation phase, and hopefully from 2025, 26 onwards, there will be an operational phase where all of these technologies will be available then for the wider community. These are the different services which I jump over, and I hope coming to the end of the presentation that you somehow got a feeling what kind of phenotyping is there, how we have to cross borders, how we can also open new windows for science, for crop production and breeding. And I hope that there are some questions and I didn't talk you into too much trouble. Thanks a lot.
Thank you, Dr. Schurr. That was an excellent presentation. An excellent presentation, and we do have a number of questions coming in.
I see you talking, but I can't hear you.
Okay, just a moment.
Try again.
Yes, can you hear me?
Yeah, now it's good. Yeah.
Okay.
Now
Okay, great.
thanks.
Okay.
Yeah.
Thank you so much for an excellent presentation. That certainly opened, I'm sure, a number of eyes into the wonderful capabilities that this technology provides. And yes, we do have a number of questions that are coming in. So we'll try to get to as many as we can. Just a reminder to all of you, we noticed that some of you are putting some questions in the chat feature. If you could go ahead and please put those questions in the Q&A panel. That way we can capture your questions, and if we happen to not be able to get to your question live, then we will certainly follow up with you and ask Dr. Schurr to help us get your questions answered. So let's get started. And so the first question, Dr. Schurr, is how can we use this innovative phenotyping methods in on-farm experiment programs with farmers?
Yeah, that depends obviously in many different backgrounds. It depends first on what kind of crop you're talking about, because there's a wide range of different crops, obviously. There is a lot of activities where we are talking to groups which are actually translating this into day-to-day useful things for farmers. There's a lot of activities with ag industry, So in many cases, there is a need, still a need to get some expert knowledge on the ground. But many of the technologies are actually available in a way that this is more and more now also going into an open source information, which can be used for farmers on site. But it very much depends on the situation. So what kind of crop, what kind of diversity of crop, what is the question, what farmers want to use with that. So there's a lot of discussion there, how to actually develop that specific approach. But it's definitely the target is to actually bring it to the farmers and to the breeders.
Okay, great, thank you. So the next question is in a comment as well. Thank you for the excellent and informative presentation. I would like to know if you have perhaps conducted any collaborative work in sugarcane for the purposes of breeding, and if so, what successes or challenges have you experienced with implementing the technology in general?
So in Germany, we don't grow sugarcane, and therefore we did this in actually international collaboration. We have done intensive international collaboration with Brazil over the last 10 years or so. We had very strong interaction there with Embrapa, who themselves also built up phenotyping facilities for sugarcane. In that specific case with sugarcane, because we can't grow it here in a temperate situation, we have more developed technologies together with the colleagues there because we have technology development expertise. And then they were taking that further into the field and further into the interaction with the farmers, with the breeders, etc. So in our specific case, yes, we worked with sugarcane, but it's not our head crop here.
And would you like to discuss any challenges that you've experienced with the technology?
The size of the plant is an issue because it's growing very quickly. So therefore, you have to adapt your systems to the field conditions in the tropics. We also have quite a number of other tropical phenotyping activities, which we presently do with Thailand, for example, on cassava. Or with Taiwan or with Africa on cassava. This is not sugar beet, but the tropical conditions are challenging for a lot of technologies. But this is, as sorry to say again, as in the thing before, it very much depends on the situation on the ground. And in these specific cases, we actually use our technology expertise, but only the dialogue with people on the ground there, because they know what they want to measure and they know the specific conditions there.
Great, thank you. So the next question is, how effective is spectral imaging for precise disease screening for pathogens such as Puccinia strophiformis?
So we're working quite a bit with different spectral information there and obviously there's a lot of hope that spectral information can give us information earlier before we can visibly see things on disease. We are not people working on a specific disease, so we are providing our technologies together in the dialogue together with colleagues there. There are some examples where we can see before visible symptoms come up that there is a disease coming in, and that would allow to spray before things happen, and also reduce the amount of spray needed and being more specific there. On the specific disease, I don't have any experience, but it would very much depend, as I said, depending on the conditions, interaction with people who have that technology in hand. And we decided here that specifically on disease side, we only do disease work together with people who know their biological system, because we are not the disease experts, the pathologists, but we have technologies to do that.
Okay, thank you. The next question is about training. And so are you providing any type of training? And so we have someone that's currently working on multispectral images captured by Centera double 4K multispectral sensor integrated on the DJI Inspire 2 drone. So do you do any type of training?
That sounds like a interest in a very specific training for a very specific setup with the specific camera and the drone. We can try to do that. That's not our major target to do individual trainings there for a very specific thing. But if people have a very specific question, the email is out there, so people can contact me on that, and I would pass that on to the colleagues here. Or to any other colleagues in the networks, because the networks are very important to also hand out these kind of questions to the right people. What we do is we have specific training sessions in general for phenotyping for different technologies. We also provide access in teaching people how to use technologies here on site, but also in the European platform. And there's, besides the European platform, actually, there's also the International Plant Phenotyping Network, which I'm still the president of, which is also providing training and providing access to this kind of technology. So there is a global network of people knowing how to do that, and we use the network in order to guide people to the right spot.
Okay, great. So the next question is about artificial intelligence. Do you think that AI will have an increasing role in better understanding phenotyping and the link it provides for gene function?
So AI is common practice, I would say, in many fields of phenotyping, which are using images or a high number of data today. We have people working specifically on AI technologies. There's quite a lot of communities out there. At the same time, the phenotyping community itself is trying to link up with the dedicated AI community, which is so that we don't have to reinvent the wheel from scratch. So there is quite a lot of things happening at the moment. Actually, within the IPPN, we are presently working towards a working group which is specifically focusing on AI, to bring together AI experts with people doing specific technologies and with other people who want to use phenotyping for their biological questions. The second part of the question, actually, if AI would help to integrate between phenotyping and the genomic basis, that obviously depends on specific setups of experiments and data collection, which includes the information available on the genetics, as well as the information available on the phenotyping. I'm pretty sure there will be, but this is not what we are specifically working on at the moment, but this is more kind of community interaction between the genomics and the phenotyping community.
And probably our last question that we'll have time for today, is there any current collaboration with wheat breeding?
Oh yeah, there's a whole wheat phenotyping community. There is which is actually from the Wheat Initiative. So the Wheat Initiative has, which probably many of you know, has a number of different sections. When this Wheat Initiative was set up, I was actually co-leading the wheat phenotyping group there. I handed this over, I think, 2 years ago now to other people. The whole setup changed a little bit, but the Wheat Initiative has a whole wheat phenotyping community, which specifically looks at wheat. And I would very much recommend people to link to the Wheat Initiative, and people can apply to become a member of that wheat phenotyping community there. Yeah, that's a direct link there. And there's a lot of wheat phenotyping out there.
Okay, Wonderful. Well, there are a number of questions that we will not have time for today, but what we'll do is we'll provide a list of those questions to Dr. Schurr and help him maybe answer. But he also provided a email for direct contact, so we encourage all of you to do so. If you would like to speak with him directly. So once again, Dr. Schurr, we thank you for joining us today. This was an excellent opportunity to talk about a very popular topic, as you can tell from all of the questions. And thank you everyone for joining us, and we hope you have a wonderful day. Thank you, Dr. Schurr.
Thanks a lot.